Introduce the next speaker, or the next discussant rather, who is Dr. Jennifer Ricker from the University of Colorado, who will be discussing the next 5 posters.
Okay, if the slides will get started, I'm happy to be here today and summarize these 5 excellent posters. I'll give a brief introduction of how I think that they tie together once these slides start. I'm Jennifer Ricker. I'm in the Department of Pathology at the University of Colorado Anschutz Medical Campus. And I have a poster that's going on right now, so I'm missing that to be here with you for the spotlight presentation. So we can go ahead and skip on to the next slide since I already introduced myself. And so today, all of these posters are on triple-negative breast cancer, which, as you know, they lack HER2 overexpression, and they lack estrogen and progesterone receptor. And really, what all these posters have in common is that they're trying to find new vulnerabilities of triple-negative breast cancer that we can target. Because other than chemotherapy, there's not, and now immunotherapy to some degree, but we still need more targeted therapy approaches for triple negative because, as most of you know, they have a rapid recurrence within the first few years of diagnosis. So the first poster is talking about the luminal androgen receptor subtype of breast cancer. And one reason it's important to target this subtype is because they have one of the poorest pathologic complete response rates to chemotherapy, as can be seen here in this nice paper by Naoto Ueno, and others have recapitulated this finding. So it would be good to find another approach for the luminal androgen receptor subtype of triple negative. So as I said, that poster will come first on tackling the luminal androgen receptor subtype, and we're applying some lessons learned from the ER positive subtype, but what about these non-luminal triple negative breast cancers? So the next poster will explore the role of the Sark family, Sark family kinase YES1 in basal-like triple-negative subtypes. And targeting YES1 disrupted chromosomal stability, leading to increased double-stranded breaks. The next poster will examine the interactions of the DNA damage repair pathway with the PI3 kinase pathway activation. And new generations of inhibitors are now have been developed with less toxicity and different patient selection criteria, and that makes it worthwhile to reinvestigate these inhibitors of these pathways and new inhibitor combinations. However, as I was saying, what we lack in triple negative are novel targets that represent unique vulnerabilities. So the last 2 posters explore differential roles of 2 small GTPases, RAL-A and B, and the last one is a very interesting approach examining other potential reasons besides reducing bone fractures that bisphosphonates provide a survival advantage in triple-negative patients, breast cancer patients. So we'll get started with the first poster, which is by Gunmin Kim, and he, working with Carlos Arteaga's group at UT Southwestern. Oh, let me go, well, that's okay. So as I said, they're looking at the luminal androgen receptor subtype, and what other groups have also found is that the PIK3CA mutation rate is higher in the luminal androgen receptor subtype than in other subtypes. However, RB loss or deletion is less common. So they're looking at potential therapeutic targets for the luminal androgen receptor subtype and looking at CDK4/6 inhibitors, PIK3CA inhibitors, and androgen receptor targeting drugs. And the purpose of the study was to investigate which one of these combinations, alone or in combination, would be most effective in this subtype. So they did cell viability assays and showed that RB intact LAR triple negative lines are sensitive to single agent palbociclib, I can never pronounce it, alipicib, and I can never pronounce that one, and also an AKT inhibitor, which I will not pronounce either. And then they looked at clonogenic assays of 2 LAR lines treated with palbo-alpelisib and capivasertib and enzalutamide. I can do that one. So what they found was, to a certain extent, in these LAR subtypes, but not as much in the other subtypes that are represented here, MCF7 and BT20, these drugs are somewhat effective and actually more effective than enzalutamide at the doses shown. So the take-home message of this poster, and I'm apologizing in advance that I have to skip through these so fast in the interest of time, but yeah, the take-home message of this one, hopefully you got to visit with the poster presenters and talk in more detail. But the CDK4/6 inhibitor, which suppresses RB phosphorylation, can induce AKT activation. So in addition, addition of the AKT inhibitor to palbociclib suppresses this rebound activation of AKT that follows the CDK4/6 inhibitor. So AKT inhibitors could be a better combination partner for Pablo than PI3 kinase inhibitor in RB intact LAR triple negatives. So going on to the next poster, this one is from Dr. Ruth Curie's lab, Katrina Piemonte, and they're at the Cleveland Clinic. And the title of the poster is "The SRC Family Kinase YES1 Controls Chromosomal Stability in Triple-Negative Breast Cancer." Oh, we're missing some. Here we go. So YES is highly expressed in triple-negative breast cancers, the non-luminal subtype, and associates with poor outcomes. So here you can see YES immunohistochemistry. It's higher in triple negatives in general than the ER positive, HER2 negative subtype, and you can see quantification of that over here. And also it's associated with poor outcomes, and this is in all subtypes, and recurrence-free survival and overall survival. This slide shows that YES1 is necessary for triple-negative breast cancer growth and viability, and they're showing this data in MB468 cells. They've knocked down YES1 by siRNA and showed that, and silencing was confirmed here. And they show that loss of YES1 causes growth suppression and induces apoptosis. And this data was repeated in 2 other triple-negative cell lines. On this slide, you see that loss of YES1 results in a delayed cell cycle progression and chromosomal instability. So we have cell cycle analysis by flow over here in A, and the chromosomal instability-associated genes were enriched following YES1 silencing, and triple-negative cells with YES1 knocked down developed abnormal nuclear phenotypes associated with chromosomal instability. So in summary, mitosis and DNA damage regulating pathways are novel downstream effectors of the YES1 SRC kinase family in triple-negative breast cancer. And ongoing studies will identify the proteins that are phosphorylated by YES1, discover gene networks, particularly those in mitosis and DNA repair that are controlled by YES1, and assess the impact of combining selective YES1 inhibitors with DNA damage-inducing agents for potential synergy. So stay tuned for that story. The next poster is out of Sioux Falls, South Dakota. The title is "Linking Proliferative Signal to DNA Damage Signals in Tumor Cells, a Contextual Synergy Between the PI3 Kinase Pathway and the DNA Damage Response Pathway in Triple-Negative." So they examined alterations in genes in the PI3 kinase and DNA damage repair pathways from 93 triple-negative breast cancers and did string analysis and network summaries. to look at interactions in the protein products of these genes. Triple negative is commonly associated with oncogenic alterations of these two pathways and represented by the loss of PTEN function or activating mutations in the PI3 kinase and loss of BRCA1, reflecting DNA damage repair. So they used cell line models and to test the cooperative effects of different drugs that target these pathways. They tested apoptotic signals, proliferative signals, mitochondrial potential with a TMRE-based assay, the clonogenicity assays, and this was all following single and double triple combinations of inhibitors using isoform-specific inhibitors of the PI3 kinase pathway, the AKT inhibitor, an mTOR1 and 2 inhibitor, and in combination with PARP inhibitor, talazoparib or olaparib, plus a DNA-damaging agent, carboplatin. So really what they're trying to do is tip the balance towards apoptosis and triple negative. And the summary of this poster is that they see that combinations of targeting these two different pathways together may be effective. And there's not time to walk you through the copious data, but the take-home message is that the PI3 kinase pathway cooperates with the DNA damage response pathway, and inhibition of both pathways is more effective than either alone to inhibit proliferative signals and induce apoptotic signals. Moving on to the next poster on investigating the role of the paradoxical roles of these two different RAL-A and RAL-B in triple negative, and Jonathan Spihar out of the Ohio State University in Columbus, Ohio, was the presenter of this poster. And it's quite interesting because even though they're closely related, they act quite differently as the bottom line here. RAL-A and RAL-B are small GTPases with quite a bit of homology. These RAL-A isoforms share GEF and GAPs and downstream effectors. Despite their significant similarities, RAL isoforms have a disparity of function in several other cancer subtypes, and the goal of this work was to test the requirements of RAL-A and RAL-B in triple negative growth and metastasis. So as you can see here, RAL-A promotes, but RAL-B inhibits triple negative tumor growth. So CRISPR knockout of these two isoforms in the 231 cells, as confirmed here, showed that RAL-A knockout significantly reduced, but RAL-B knockout significantly increased growth of MDA231 primary tumors in non-SCID gamma mice. Again, RAL-A, but not RAL-B, promotes lung metastasis in triple negative as well. So a comparison of lung metastatic burden in mice with equally sized primary tumors that were adjusted for that shows that RAL-A knockout significantly reduced the metastatic burden, but RAL-B knockout tended towards increased metastatic burden. So RAL-A expression is prognostic of breast cancer patient outcomes, and that's what's shown here with immunohistochemistry staining and outcomes data as well. So in summary, these have quite different functions. RAL-A, but not RAL-B, is associated with increased triple-negative tumor growth and metastasis in vivo and poor outcome in triple-negative breast cancer patients. So future studies will address the molecular basis for these disparate roles in triple-negative breast cancer. Moving on to a look at the enzyme CD73, this poster is quite interesting. It's looking at how a bisphosphonate, a certain type of bisphosphonate, induces a B and T cell accumulation in triple-negative breast cancer. And this was by a group, Natalia Petruk, in Finland. So here, bisphosphonates inhibit osteoclasts and prevent bone fractures. And in particular, the nitrogen-containing bisphosphonates have immunologic effects, but the significance of these effects remains unknown. So when given in the adjuvant setting, bisphosphonates provide a survival advantage by reducing bone fractures, but they think that something else may be going on here. So the hypothesis is that CD73 may affect immunological bisphosphonate responses in breast cancer. And you can see some of the data here that I'm going to kind of cut to the chase here so that we don't go over too long. But they have in vitro and in vivo data on how, in the presence or absence of low versus high CD73 expression, how this particular bisphosphonate works. So again, let me just skip to the take-home message. Hopefully you can go back and talk to the poster presenters and ask some questions here as well. But the bottom line is that these immunologic effects of bisphosphonates, rather than directing direct growth of inhibitory effects, and this may explain why these drugs provide survival advantage to some breast cancer patients. The tumor characteristics such as CD73 and other tumor immunity affecting proteins may explain variation in the bisphosphonate responses between patients with low versus high CD73 expression. So the next step is to investigate whether B cells can contribute, because they saw more B cells, as you can see here, with the low CD73 expressors. And they will test how this contributes to the bisphosphonate treatment responses in breast tumors and how the tumor growth in general is affected. So yes, B cells play a controversial role in breast cancer, and this may help elucidate how bisphosphonates are acting to improve outcomes in addition to their usual purpose, which is to reduce bone fractures. So thanks for listening to my summary of these 5 excellent posters, and I hope you have questions for the investigators themselves, because I'm sure they can answer the questions even better than I can. Thanks for your attention today. I'd be glad to take